Hello, everybody. In this lecture, we're going to talk about transcription. So let's get right into it. By the end of this lecture, you should be able to explain protein synthesis, aka gene expression, describe how it impacts phenotype, and state why it is essential to cell survival. You should be able to explain transcription and not as much translation in this one. We'll talk more about that in the next lecture. Point out differences in the process between prokaryotes and eukaryotes. Define reverse transcription, describe structures and functions of the three main RNA types that we're going to discuss, and summarize splicing of mRNA in eukaryotes. So let's start out with just some basic explanation. So protein synthesis, which is what is necessary to actually express genes, is when we take genetic information within a cell and we create some sort of gene product. To do that, we have to first copy the DNA over to RNA through a process called transcription. And then those transcripts are brought to the ribosome as messenger RNAs, and they build proteins through a process of translation. So to kind of summarize that, the gene is in the DNA. And when it needs to be expressed, it will be transcribed to a messenger RNA, and that messenger RNA will be translated to a protein. So protein synthesis is central to life, and it is going to be how you get most of the physical and functional features of cells. And an example of this that we've touched on multiple times throughout class is that many of the antibiotics that we use to fight infections work by interrupting protein synthesis. Now, there are three steps to transcription, which is the process of making RNAs. In eukaryotes, transcription is going to occur in the nucleus, and in prokaryotes, it's going to just occur in the cytoplasm. Now, there are going to be three steps. That's initiation, elongation, termination. So this is similar in terms of DNA replication. So one thing to keep in mind is which strand is going to actually be transcribed. So the strand that is transcribed is called the antisense strand. The antisense strand is going to be very similar to the leading strand in DNA replication. So transcripts are only made continuously. They are not made discontinuously. And so that's what we see here. So this is an RNA polymerase adding the mRNAs, adding the RNTPs to make an mRNA, and it's doing so continuously as the strand unwinds. And so when we think about DNA replication, we don't use these terms sense and antisense, but we do when we're thinking about transcription. And so the antisense strand is going to be the 3' to 5' strand. When you open up that, when you make that replication fork or that transcription fork or transcription bubble that we'd be making here, the antisense strand is going to be read in that 3' to 5' direction. This is the same way that all the polymerases read the old strand. And so the old strand is oriented 3' to 5', and then the new strand is going to be added 5' to 3'. And so it's important that you understand that distinction. So be sure to kind of look at this image and get what I mean. When we're reading the strand, that means that we're reading the old strand. And we're going to say that, okay, this is an A, and then this is a T, this is a G, whatever. And then we're going to polymerize the new strand in a 5' to 3' direction. So that's the bottom strand here. And in this case, this bottom strand is going to be mRNA. Now, the sense strand is not transcribed at all. That's the one down here. And it's nearly an exact match of the transcript, right? So the only difference between this transcript and the sense strand is that the transcript is going to be made out of RNA bases, the sense strand is going to be made out of DNA bases, and the transcript is going to have uracils while the sense strand is going to have thymines. So an initiation, we have a formation of a transcription bubble, very similar to the initiation of replication. Your topoisomerase or gyrase is going to unwind the supercoils of the DNA, right? And then helicase is going to open that up. Now, RNA polymerase needs to find these specific sequences in the DNA that are known as promoter sequences. And they are in bacteria in particular, which is what I'm going to discuss here. You have these -35 regions and these -10 regions. So the sigma factor is going to be part of your RNA polymerase. It's going to be looking for these different sequences that are going to be upstream of the transcription start site. So the sigma factor is going to actually bind to that -35 region of the promoter. And those two regions of the promoter are pretty highly conserved, which means that there's not a lot of change in those bases over time, and there's usually going to be a lot of carryover between species that have the same genes. And that's because those sequences are highly selective for the promoters. And if they're not getting good contact with the promoter, then they're not going to get made. And then the gene's not going to survive. And if that gene is necessary for life, then the whole organism isn't going to survive. You may see the term teta box or tata box referring to that -10 region because you have this TAA motif. So once polymerase is bound, once it finds its proper promoter, it's going to begin replicating the DNA or transcribing the DNA to RNA. And it does this for all types of RNA, but when we're talking about gene expression, we're specifically talking about mRNAs. And so it's going to read the antisense strand, 3' to 5', but it's going to make the new strand 5' to 3'. So this is exactly the same as DNA replication. There's nothing different here. The only thing that we can consider being different at all is just the fact that we're making a transcript instead of a new DNA strand. So RNA polymerase is the same type of enzyme as primase, and so they don't need any leading bases to begin transcription. They can just start adding those RNA bases right away. And then the binding process is U to A and G to C. In this case, U to A has 2 hydrogen bonds and G to C has 3. So eventually transcription is going to stop though. So unlike DNA replication doesn't stop until you go through the entire genome or through the entire chromosome. And transcription, you stop when you actually get to the end of the gene. And so there are two mechanisms that we can briefly talk about. One is called row-dependent termination. And in this, you get this protein called Rho, which is like this hexamer, and it travels behind the RNA polymerase, and it's wrapped around the transcript. So you can kind of see it. It's following that RNA polymerase down there. And as it's coming up, the DNA is kind of or the DNA and RNA bonds are being broken, those hydrogen bonds. And RNA polymerase is proceeding in that 5 to 3 direction. And eventually the polymerase is going to have this string of guanines. And so if you remember, guanines have three hydrogen bonds, and so they actually are a little bit harder to make. And when this happens, when you get a string of them, you slow down the polymerase, and so it begins to stall. And so then uh, that row is going to actually catch up and bounce the RNA polymerase off of the DNA. And so essentially what happens here is that when you have row-dependent termination, the RNA polymerase slows down, and then the row bumps it off of the DNA. Then we have row-independent termination. In this case, we are going to actually terminate based on structure that's being formed by the RNA. And so here, there's also a GC-rich region of the transcript that causes this formation of a hairpin. So one of the cool things about RNA is that it's single-stranded, but it almost always has secondary structure capabilities. And so you get these GC-rich hairpins. So basically, this single-stranded RNA folds over on itself to make the secondary structure. And then you get a U-rich track, which is going to mean that you make a bunch of A's, right? And so when you hit that AT rich region, you're going to make a bunch of U's. And so this is going to be weaker hydrogen bonds because there's only 2 hydrogen bonds there. And that hairpin is actually going to put strain on that weak U-rich tract. And that U-rich tract is then going to cause the RNA polymerase to stall up and break loose from the DNA. And when RNA polymerase is bounced off that DNA, transcription stops. And so this is a little bit of a summary of what's going on. You've got initiation, where RNA polymerase binds the promoter, DNA unwinds, revealing your template strand. RNA polymerase is going to elongate in the 5-3 direction, adding complementary nucleotides. And then you get termination. You're going to hit a sequence that basically causes RNA polymerase to slow down. And in doing so, the RNA dissociates from the DNA. So generally, when we think of transcription, we go from DNA to RNA, although there is some capability of reverse transcription. Viruses like HIV have reverse transcriptase. Also, some cells could use this as well. And this is where RNA is used to make a template DNA or a copy DNA, which we call cDNA. And that enzyme that does that is reverse transcriptase. So really what I want you to just know there is the ability to go from RNA to DNA, which makes cDNA, is known as reverse transcription and uses reverse transcriptase. So we have 3 major types of RNAs that we'll talk about here in gene expression. That's mRNA or messenger RNA. That's tRNA or transfer RNA, and rRNA or ribosomal RNA. And so the mRNA is going to have the codons that code for the amino acids and for the stop codon that stops translation. And so the way to think of this is that the mRNA carries the message, which is the code for making the protein. So the tRNA contains an anticodon, and it's complementary to the codon. So codon binds to anticodon. And the tRNA recognizes that codon on the mRNA, but it brings in an amino acid as well. And so it brings that amino acid into the ribosome, and it attaches it to the growing amino acid chain. And the rRNA has this 3D structure that combines with your ribosome, and they have catalytic functions, so they help actually process and make these these ribosomes work properly. So here's kind of an example of those, like in a picture format. You've got your mRNAs with your codons. You got your tRNAs that have a codon, an anticodon on one end and a amino acid on the other. And then you have your rRNAs, which are combined with your ribosomes. Eukaryotes have a special process of splicing mRNAs, and they have to be transported out of the nucleus before they can be translated. And so in the splicing of RNAs, there are sequences that the final transcript don't need, and they are clipped out. And so you've got two types. You've got exons and introns. So when you transcribe out your gene, you're going to have exons and introns. And so the exons are segments of mRNA decoded to build a protein. And then introns are intervening sequences of mRNA that are not decoded to build the protein, right? So we keep the exons and we cut the introns. And so mRNA splicing has the potential for having alternative splicing that gives you different combinations of exons. So you can actually pack more information into the same sequence, so you don't have to have independent genes for each of those. And so here's an example where you have a eukaryotic mRNA where you have these introns and exons. And then the spliceosome comes in and it's going to cut out the introns. And then you keep your exons 1, 2, and 3. They get joined together, and now you can translate that. All right. So here, you should be able to now explain protein synthesis, explain transcription, and to some extent translation, although we're going to talk much more about translation in the next one. Define reverse transcription, describe the structures and functions of the 3 types of RNAs, and summarize mRNA splicing in eukaryotes. All right. Hope you enjoyed. See you soon.